Welcome, everyone. Good afternoon, and thank you for gathering with us today, both in person and virtually. Before we begin, I'd like to acknowledge the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within the Suquamish, Tulalip, and Muckleshoot nations. It is my honor to welcome you to our annual Omenn Lecture in Environmental Health, sponsored by the Department of Environmental and Occupational Health Sciences, as well as the UW Microbial Interactions and Microbiome Center, which I'm told is called MIMSI. This lecture honors Dr. Gilbert Omenn, former Dean of the University of Washington School of Public Health and former chair of DEOHS, who is here today with his wife, Martha Darling. Doctors Omenn and Darling are both generous supporters of our school and the department. Thanks to Gil and Martha, we have recently updated our environmental health research laboratories, making a transformative impact on the most important work DEOHS scientists do. Please join me in giving Gil and Martha a round of applause to thank them for their support. Our speaker today is a hero to many of us in this room across the University of Washington campus, the nation, and the world. Dr. Rita Colwell earned her PhD in marine microbiology right here at the University of Washington. Go Dogs. Woo-hoo! Through her innovative research and 40 years of scientific leadership, she has helped define our current understanding of infectious diseases in the environment, particularly cholera, which she's going to talk to us about today. She has also developed advanced technologies to stop the spread of those diseases. Dr. Colwell now leads an international network to address emerging infectious diseases and access to safe drinking water. Her list of awards, achievements, and service to the scientific world is very long, so I'm going to name just a few of them. She served as the 11th director and first female director of the National Science Foundation. She is the founder and chairwoman of Cosmos ID. She has authored or co-authored 17 books and more than 800 publications. She is a member of the National Academy of Sciences and has served as the president of the American Association for the Advancement of Science. She holds, get this, of all those things, this is one that was like, what? She holds 62 honorary degrees. And last but not least, there is a geological site in Antarctica called Colwell Massif, which is named in recognition of her work in the polar regions. She currently serves as Distinguished University Professor at the University of Maryland at College Park. Please join me in welcoming this year's Omenn Lecturer, Dr. Rita Colwell.
Thank you for the extremely kind introduction. And Gil and Martha, thank you so very much for the opportunity to come back to the University of Washington, my alma mater, doctoral alma mater, and to be here to be able to speak to you about the last 60 years of research. So for every minute of this discussion today, it's 1 year of research. And I've deliberately shown the slide of the Vibrio cholerae, Vibrio vulnificus, Vibrio fluvialis, and it goes on. There are now 200 species. When I was a graduate student starting my work here, there were 3. Now there are more than 200. And CDC, Centers for Disease Control, has decreed that all species of Vibrio cholerae infections have to be reported. So Vibrios are known pathogens. We all have lived through the COVID pandemic, but I want to emphasize we were simultaneously undergoing 2 pandemics. The problem was that the 7th pandemic flow of cholera was underway at the same time that we were dealing with COVID, but it was mainly in the developing countries or the lesser developed countries that the cholera epidemic was continuing. Pandemics, we can anticipate, will continue. We are not past them. And it certainly is necessary for you workers here in the School of Public Health and all of us to make sure that vaccine research and vaccine development, distribution, availability continues. I'm going to talk about Vibrios, mainly Vibrio cholerae, but the work began when I was a graduate student at the University of Washington. The study that I did was to look at the bacteria associated with marine animals, particularly oysters and various important that are associated with marine animals and the aquatic environment. And at that time, this was way back in 1960. At that time, there were probably 2 or 3 species of Vibrio. One of the early species was Vibrio anguillarum, which is associated with eels. And the subsequent work that I did as a postdoc in Canada at the Research Council was studying more intensely the systematics, finding that the type species for the genus Vibrio was Vibrio cholerae. And so the association of Vibrio cholerae with these aquatic environmental bacteria was not an acceptable hypothesis. And I had to struggle both as a woman scientist, because I think if I'd been male, I probably would have been able to change minds a little earlier. But in any case, it was deemed by the medical community that cholera was caused by a bacterium that was transmitted person to person. Well, the work that began at the University of Washington was that Vibrios were common to the aquatic animals, and in particular, the bacterium which caused cholera had been associated specifically with human infections. Now, cholera is an acute water-related diarrheal disease. It's transmitted water and food. It's reported even today in about 50 countries and affects millions of people. But and new biotypes have emerged as well. We cannot eradicate the cholera bacterium because some of them are bioluminescent, some of them fix nitrogen, and they certainly are involved in carbon cycling. So they're part of the natural environment. And I just want to emphasize that in case of cholera, today, the most recent data were for 2023, when I looked it up, and 50 countries were impacted, 700,000 cases, 4,000 or more deaths, and especially for infants, if not treated within 4 or 5 hours, death ensues. So the work that I continued after studies in Canada in the Chesapeake Bay was the discovery that really the vector, the host, the vector for the cholera bacteria is the copepod. It's a microscopic animal widely distributed in the world oceans, and the most common component of the planktonic composition of aquatic systems. We developed the first model for the transmission of cholera to humans, working out the environmental cycle. First in Chesapeake Bay, for example, and certainly in the water areas around Seattle, and in the Gulf of Mexico. I didn't say the Gulf of America, I said the Gulf of Mexico. Yes. Mea culpa, mea culpa. We found that the bacteria in the spring months, the sunshine increases, the water temperature goes up, the phytoplankton respond, and then weeks later, the zooplankton will have fed on the phytoplankton, and their bloom will crash, and that releases the Vibrios into the water column. In countries like Bangladesh, where water is drawn directly from the streams for drinking, then the ingestion of the cholera bacteria then does its work. Work that was done at the University of Maryland in the medical school, not by my team, but about 60 or 70 years ago, were volunteer studies which showed that if volunteers take in 1 million Vibrio cholerae per milliliter or per teaspoon, if you will, then you get cholera. Less than that, you may just have mild diarrhea or vomiting, but not the full-blown disease. In any case, the transmission then can be environmentally mediated. This work was done way back in 1975 And I assure you, it was not accepted. Clearly, they did not accept that the Vibrio cholerae could be in the environment. And it took some really intensive experimental work. This was done in 1970 with one of my graduate students, Ron Citarella, where we did that very tedious, this was early DNA work, very tedious hybridization where you separated the strands of the environmental Vibrio cholerae and they separated the strands radio labeled of the pathogenic from a patient Vibrio cholerae. Then you hybridized through density gradient centrifugation, which took 24, 30 hours. And then you would get three bands. You would get the environmental banding, the clinical, and then the hybridized of the two. It was clear, and that was published in 1970, 71 or so, that the environmental Vibrio cholerae and the clinical were really the same species. They were genetically very similar. I mean, that sounds similar, silly to you today, but I must tell you, it was painful. We got it published, but still, the community was not happy with us. Some of the more recent work, I think, has been pretty dramatic because what we have been able to show is taking advantage of the fact that more than 50% of all us human beings live within 50 miles of the coast. Some work that we did in collaboration with the marine labs in Southampton and Plymouth, and in collaboration with my colleagues at the University of Genoa in Italy, Dr. Carla Pruzzo and her team, and the Max Planck Institute, where they did a good deal of the DNA work. We took the samples that the Plymouth and the Southampton labs had been collecting for about 40 years in a form of citizen science. What they had done was to send these plankton collectors to ship captains, yachtsmen, to ferry boat captains around the North Sea and asked, just collect some plankton, send it to the laboratory, and just give us the GPS location. And what we did was this 40 some years of collected plankton, we extracted the DNA and we were able to detect using PCR at the time that we did this work, and being able to detect Vibrio cholerae, Vibrio vulnificus, a pathogen, and Vibrio perihemolyticus, another known pathogen. And we were able to show that with the temperature increase over that 40 years of the sea surface, we were able to detect there was a determinable increase in the numbers of these Vibrios associated with those plankton samples. And we also were able to correlate this with the increased incidence of vibriosis, Vibrio infections, not so much cholera in Europe and in the northern part of the US, but Vibrio perihemolyticus and Vibrio vulnificus. So we published this in PNAS, and even, I should say that even with this correlation, there still remain some dispute about whether in fact this was really associated with phytoplankton. So we did the following experiment. We were able to, we made a proposal to NIH, to the NIAID, to do a filtration experiment. That is to go in to the villages of Bangladesh where we had started our work in 1975, and we were proposing this about 1980, to have filtration done using sari cloth, which we had tested in the laboratory to show that we tested all kinds of material, men's t-shirt material, used sari cloth, new sari cloth. And in the lab, we showed that if you folded old sari cloth about 4 or 5 times, we looked under the electron microscope and it showed that clearly you got a 20 micron mesh filter, which would be suitable because the plankton are about 200 microns and the bacteria, a few might slip through, but that earlier study showed that you'd only get mild diarrhea, you wouldn't have an outbreak of cholera. Well, the NIH said, no, that's too simple. They rejected it. And so we then asked the Thrasher Foundation if they would give us money to get some data to begin with, and then we'd go to NIH. And we did do that. We were able, with a Thrasher funding, to go into the villages and to demonstrate that you could reduce the number of Vibrios by this simple filtration technique. And then we applied to the NIH, and the NIAID said, well, this is not sophisticated, but the Nursing Institute will fund it, which they did. So we were then able to carry out a 3-year study, about 100 villages, thousands of participants, and we were able to demonstrate that by this technique, we could reduce cholera by about 50%. It was pretty clear to the women, who were the ones who collect the water for the families during the day, that when you had nice, clear water, that it was a lot better for your children than the water with things swimming in it. So that study showed very clearly we could reduce cholera by 50% by this very simple technique. And I think that was the beginning of the acceptance that the plankton were the vector, that the bacterium Vibrio cholerae does exist in the environment. And what we also found out was that, I should go back just a minute, and we found out that when you looked for the Vibrios in the winter and you did the culturing techniques, they didn't grow. But we developed a monoclonal antibody that we ligated to sodium isothiocyanate, which, when you look at the stained bacteria under ultraviolet microscope, they glow green. And so we were able to demonstrate the presence of these bacteria when you could not culture them, particularly during the winter months when the temperature goes down. It turns out, because their host, the vector, the plankton, at least in the Chesapeake Bay, in the wintertime go into a diapause. They dive down into the sediment. They overwinter in this diapause stage. And the Vibrios, their bacterial friends that live with them, the commensals, do the same thing. They go into a viable but non-culturable stage. And again, that was contentious. The CDC, some of the scientists referred to them as Colwell's ghosts. But we now know that this is a phenomenon that many, many other bacteria, particularly gram-negatives that don't form spores, undergo, because most of the gram-negatives, at least the waterborne ones, don't need to form a spore coat, as those that are found in soil, like the bacilli and the clostridia, form a spore to prevent desiccation. So in any case, that's another aspect of the discovery. In addition, it occurred to us that if the water temperature and the relationship with phytoplankton was so important, we could measure this by satellite. And so some 25 years ago, we did just do that, because the NASA had launched Landsat, a satellite that measured the various types of chlorophyll, A, B, C, et cetera, sea surface temperature and sea surface height. So a team at NASA and we joined forces, and we did a series of studies which showed that you could use the satellite data and you could predict the risk of cholera, because the conditions then would fit that we had shown were associated with a bloom of the plankton, the then successive bloom of the bacteria, and then the correlation with cases of cholera. So shown here are the data that we published some 25 years ago, showing the red line are the cases in the Bay of Bengal, in the Sundarbans, the area right along the bay. And the blue line is just one of the parameters, is the temperature. And that was very crude. It was a mile quadrant measured by satellite. And so the relationship of the temperature and the chlorophyll allowed us to be able to predict when and how aggressive the cholera outbreaks would be. We got a more sensitive a computational model now for this hypothesis of the relationship of cholera and these environmental parameters. And we use far more sensitive satellites currently. In fact, a number of the satellites that have been declassified where you can actually measure populations movement, has allowed us then to have a very, very sophisticated predictive capacity for the risk of cholera. We tested this in 2017 when this incredibly bad outbreak of cholera occurred in Yemen. And so we did a retrospective analysis. We took the data and said, could we have been able to predict it? And indeed, as you can see, the upper is our model, and the actual occurrence you can see in the shaded area where in fact that's where the cases occurred. So we published this kind of a short note. It was picked up by the Scientific American in December of 2017, and in January of 2018, we received a phone call from a colleague in England who asked if we would be able to provide maps for Yemen in 2018, maybe 4 to 8 weeks. We could do 8 weeks for sure then, and we promised, and we did the collaboration. And we now have reached a point where we have gone from about 65% accurate to 95% accurate. And this was tested most recently, just a year or so ago, when we began to see signals for Malawi that the conditions were right for an outbreak of cholera, and alerting the UN, with whom we have been doing research, we were able then to prepare for the outbreak. And by the end of 2023, this was the signals we were getting in June, and by August, cases were beginning to appear, but we'd already been able to locate supplies, safe water, where it would be expected the outbreak would occur. And that turned out to be a very successful application. So here's the timeline then for the first 30 years of the research, is showing very early on work done at the University of Washington, temperature, precipitation, rainfall that is, salinity. These are crude factors that are associated with the incidence of these pathogenic bacteria. We then discovered that this association with plankton was very, very important, and that we have moved on to more sophisticated ways of doing our modeling and prediction to where we work now. What's been happening in the last decade or two is that other vibrios have become a serious problem. These non-cholera vibrios, they're not in the form of the kind of dramatic pandemic that we see year after year with cholera, but when they do occur, they can be incredibly serious. Vulnificus in particular, because it's an exposure. If you have an open wound or a cut or an abrasion, and then the bacteria invade, if it goes systemic, the fatality rate is roughly 50%. So the bacterium, if not treated, say you have a wound cut in a finger, if not treated, you may end up with an amputated finger, or worse, an amputated to the elbow. And I have given lectures routinely in Maryland every year in the Patuxent region, and there's always 2 or 3 folks who are there who have been the victims of Vibrio vulnificus. What's been sort of frightening is that the infections have been increasing in the past decades. And the upper right is very interesting because 25 or 30 years ago, the incidence of Vibrio vulnificus cases would be around June, late June, maybe early July, August, and taper off in September. Now we're finding that we're picking up cases of Vibrio vulnificus in May and as late as December. So with the climate change, we're finding that the Vibrio populations are becoming much more numerous, but also seasonally spreading out much longer. And in the long-term increase of vibriosis, I think, is quite dramatic, as you can see. This is work of Michelle Morgado who did her work jointly with the direction of Amy Sapkota at the University of Maryland and my lab. And so what we are finding is that other Vibrios, just as CDC has now decreed, have become infectious agents or recognized infectious agents, Vibrio fluvialis, alginolyticus, and vulnificus. Now, Vibrio perihemolyticus is a pretty well-known pathogen described in the mid-50s by Dr. Fujino in Japan as a foodborne pathogen. And it has become now 85% of Vibrio infections reported in the United States are Vibrio perihemolyticus, associated with ingestion of improperly cooked seafood and infections of that nature. What has happened in Florida has been really dramatic, because there, as you may remember reading the news reports, in the past couple of years, the temperature off Miami has reached temperatures of 95 to 100 degrees. And the fact that you have storms coming through, stirring up sediment, creating nutrient-rich conditions, this has led to the fact that a number of deaths have occurred after the hurricanes. After Hurricane Ian a few years ago, there were 65 cases and 11 deaths from Vibrio vulnificus. You can see that path of the hurricane as it sweeps across Florida. The highest winds were located at the position number 4 there, and that was just offshore, just off in the near coastal line, stirring up the sediment. The pH changed and became slightly alkaline, and the temperature is somewhere about 95 degrees. How we isolate and enrich the Vibrios in the laboratory: high pH, high temperature, and lots of nutrient. It's selective. So we are creating in the environment selective conditions for the Vibrios to become abundant. And then Hurricane Milton comes along, and we thought we had enough, but Milton, we had even more cases and more deaths. And this is continuing. So this is a situation that I think needs attention. We're now finding Vibrio perihemolyticus cases in Alaska in the last several years. So we are changing the environment, and we have found that there's a northern case limit that's shifting. It's moving further north about 50 kilometers every year. And just last year, in October, there was a death in Rhode Island that closed all the beaches of Rhode Island. And the year previous, there'd been 2 deaths in New York and 1 in Connecticut. And this is now moving even further north as the temperature of the waters are increasing. And the increased incidence of vibriosis in the United States from the various Vibrios has been going up steadily each year. And the infections from Vibrio vulnificus can be pretty dramatic and pretty dangerous. These are folks that are surviving. Some have not survived the infections. And so these are the various pathogenic Vibrios in the United States now. And as you can see, they're all aligned along the coast, particularly, and the different species of Vibrios are increasing, so that any one of the 200 Vibrio species very likely can be a potential pathogen. We have done some of the metagenomic analyses of these samples, and this is turning out to be very interesting, because we have taken samples and we continue to do this work. This is work in progress, and I want to give credit to Kyle Brumfield, Dr. Brumfield, student who finished his PhD with me 2 years ago. He's currently a postdoc working on a project that we have just got funded to continue the work of detecting pathogens in large volumes of water. And what we're finding, and I'm a little reluctant to say too much yet, but we're picking up all of the Vibrio species, including Vibrio cholerae, off the coast of Florida. So I think it is possible to predict that we're going to have our homegrown cholera outbreaks in the Gulf of Mexico. So I think we need to be alerted to the potential of this, and the shotgun metagenomics that we have been doing show that with enrichment, we can demonstrate the presence of all of these Vibrio species. Now, I'd like to say that what we are proposing and the work we're moving into is that if we've been able to do this kind of analysis for Vibrios, why couldn't we do this for all the other waterborne pathogens? In other words, figure out those environmental parameters that are associated with their abundance, their distribution, and their pathogenic properties. And so by looking at Cryptosporidium, E. coli, Giardia, Legionella, Salmonella, etc., we should be able to determine these environmental parameters and to begin to utilize satellite sensing as a public health tool. So this may be something that you all may want to think about. And so there are lessons from the Vibrio work that we've been carrying out for other infectious diseases. And we were able to demonstrate with COVID that, in fact, this is definitely positive because we began doing some of the analyses as you've been doing here at the University of Washington for the state of Washington. We were doing in Maryland beginning in 2020, early 2020 for the state of Maryland. And using the techniques just as you yourselves have been using here of analyzing wastewater, we were able then to develop a predictive capacity for the COVID virus, and we were providing risk maps on a county by county basis for the United States and specifically for Maryland, because in this demonstration for Frederick, Maryland, a community in the northern part of the state, we were able to show our work began doing the analyses in June of 2020, and we began to get strong signals by the end of July, mid and end of July, and cases were appearing. So we were able to provide the data for the governor to determine the kinds of restrictions that should be imposed on the community. We continue to feel that these microbiome profiles of wastewater, DNA metagenomics, are a very important public health tool. And I know in the discussions I've had with some of you that you were following the same process. And I do think we need to utilize the DNA techniques on a much wider basis and to be able to provide these prediction hubs, which we continue to do in the association with our colleagues at the University of Florida. So we're working now on developing a predictive intelligence system for waterborne pathogens using these kinds of Earth observations, temperature, salinity, various other parameters built in to not only predict the incidence, this is very crude, early work, it's very clumsy, but to be able to predict not only the potential for the outbreak, but to be able to quantitatively determine an estimate of how serious the outbreak might be based on the risk scores that we can develop with this combination of work. So I'm going to conclude the talk, be happy to take questions, using the words of John Muir. When you target any one thing in nature, he, and then I inserted and she, find it hitched to the rest of the universe. Many collaborators from the International Center of Diarrheal Diseases Research in Bangladesh, where I've worked in Dhaka since 1975, colleagues at the National Institute of Cholera and Enteric Diseases in Calcutta, India, and many students, colleagues at the University of Maryland, and particularly Kyle Brumfield and Anwar Haq, and the University of Florida, former postdoc Antar Jutla. And so with that, I thank you very much for your attention and happy to take any questions. Yes.
Is the possibility of a cholera outbreak in Florida particularly concerning given the potential for it to spread across the United States. For example, if it was to outbreak in Mobile, Alabama, it may stay to that area in Alabama, but Florida has bee parks and is a major tourist destination. So would you say that it's particularly concerning that Florida is the potential attraction point of the outbreak?
I think it's more the point, and I'm being very cautious. It's more to the point that that will appear in Florida, and I don't anticipate unless everything goes to hell in a handbasket, that it would be of the type that of problem that we see in Bangladesh because of the work done in the early 1900s, chlorination, water purification, distribution to homes that was safe. So I think we have that. As long as those systems are functioning, we're protected. But I think it's important to understand that we may be getting cholera cases that are not coming from Bangladesh, not coming from India or where it is still latent. And I also would like to point out that, different topic, but we've already begun to have occasional cases of malaria in Virginia. And these individuals have traveled not outside of the country and do not have any relatives who have been traveling outside of the country. So the environmental parameters are indicating that we have a revisiting of these diseases that we have controlled. And vaccines are also important, by the way. Yes.
Yeah, that was very good to see your environmental surveillance. I know actually right now, environmental surveillance for cholera is a real interest for CDC, for the Gates Foundation, and for WHO. And they're less interested though in coastal systems where we've got marine waters or brackish waters, and more interested in freshwater systems. So they're viewing it largely as, I believe, more of a foodborne related outbreak in places like Kibera. Can you comment anything about the
Yes.
environmental factors in freshwaters?
Yes, cholera is not restricted to marine estuarine areas. Because just the formation of the United States, those who went west and drank water got cholera because there are plankton in those environmental bodies. So it's the relationship to the chitin. By the way, plankton have a chitin exoskeleton, and every Vibrio that we have tested has a chitinase. Now, why would a self-respecting human pathogen have a chitinase unless it were, in fact, what we were ingesting? So I think that the critical point here is that if I got the question accurately, is that it's understanding the change in the environment that we're making, and the fact that having access to safe water is what protects us from the outbreaks that are occurring. And being able to understand the environmental drivers is really critical to be able to control it. I hope I've answered the question. If not, I'll try again.
The issue is that environmental monitoring for the safe drinking water and things, more the surveillance of what's in the populations, and more the surveillance like we've done for COVID, but doing it for cholera in areas.
Right. Right. I think from extrapolating the cholera model for salmonella, for typhoid fever and for malaria, people are already doing it. Some work similar for malaria, but I think we can do this for waterborne pathogens. We just need to get the environmental parameters that drive them. And I should also say that we've been funded and continue to work in Chesapeake Bay to develop a predictive model for Vibrio vulnificus, because what we are already doing for the state of Maryland and hope to do for Florida, I won't get into it, but but in Maryland, we simply know that there are certain parts of the bay where the vulnificus has its optimum conditions. And therefore you can post a sign that says, just be careful. If you get an abrasion, get treated, or please don't swim here during this period of time. And so we're already developing that. with funding from NSF, but let's hope it continues. So we are working on doing just that for Vibrio vulnificus, and I think we can do it eventually. Others, we can do it for the other waterborne diseases. But what I think it is the other aspect of your question that's important is that we need to understand that our environment is critical to our health, and that as we change it, we are in fact exposing ourselves to a greater risk for these infections that, essentially were curbed, if not cured, decades ago. And that we have to think about.
Thank you.
Yes.
Yeah, just following up on what you just said, the strains of cholera that you might be seeing in Florida waters, you expressed caution, but, is there any retrospective data or retrospective samples that you can look at that tell you whether or not that's something new? Or is it possible to look 10 years, 20 years, 30 years ago?
Yes. One of the things we're finding, which is fascinating, is that a much higher incidence of antibiotic resistance, even in the Vibrios we're picking up. And that's associated to work we had done in my lab, like in the 70s, on plasmids. We had found, one of my students did a PhD thesis on the incidence of drug resistance genes on the plasmids associated with metal resistance, because we were looking at a dredged area. And so what's happening off the coast of Florida with these hurricanes sweeping through, stirring up the sediment, you've got an environment that's heavy metal rich. And so the hypothesis is that that is triggering this increased incidence of antibiotic resistance. So that's one factor. But we're working on it, and we have the data. We'll be publishing it shortly, but it's been very interesting. Yes.
Thank you for your presentation. At the risk of getting political, how much of whom is this critical information now being communicated? What are the public health agencies that are left that are getting this information? How do we get this in front of Congress? You're so close to Washington, D.C., I'm sure you go there all the time.
Don't go there. I've lived there.
How is critical information being relayed, listened to, and paid attention to, especially at this time of decimation of public health agencies?
Oh, that's a very good question that I'd like to answer sort of a deflected way. When I was a student, and those in the room who are near my age, we depended on our state health departments. When I was a graduate student, if I wanted to know something in detail about what was happening in the state, or if in one case, I had an interest at that time in Pseudomonas, and I went to the state health lab in Baltimore, and they were the source of information. We've allowed those state health labs to kind of disappear. And I think we need to rebuild them, because that's really the strongest way to control our public health system. And so I would say that that's probably one of the major things we need to do. The King County Laboratory, as I remember as a graduate student, was a place where you got a lot of good information. I don't know whether it probably continues, but I think around the country, we really need to mobilize the state health labs, and then that'll bring us back to having the kind of national system that we have. As far as doing anything about the current administration, I guess we have to do a lot of praying. I don't know how that's going to play out. I wouldn't even try to conjecture. But there's something we can do. That's my point. And I think that's where we can actually get the country to a point where it's not that we want to force vaccination, but if we can get vaccination to 90 or 95%, then we have the herd effect. By the way, I should add, one of the interesting things about the villages in Bangladesh that we did was that we were then criticized that it's not sustainable. These women are not going to continue using sari cloth. That's not going to work. So again, Thrasher gave us the money to go back and see. And we found out we had a heck of a time determining the efficacy because the control villages where we had not trained had found out and they were filtering. So obviously it works. Any other questions? Yes.
I wanted to follow up on your question, on your comments about the monitoring and the idea that when you've had hurricanes, rolling the ocean and it pulls up particulate matter that's rich with metals, and therefore you could see a signal perhaps with the metal response elements in the bacteria. But, you know, that's, and you relate it to your organisms, but you know, for some of us that look at domoic acid and other types of bacteria, other types of diatomic compounds, we're still interested in that, because remember those metal filing studies where they tried to increase the ocean efficiency and you know, and they
Right.
fed it with metal filings, and
Right.
the same thing is seen. It seems like we should learn that lesson and start to incorporate that more generally in these models for the marine, you know, the coastal areas and even mid-sea.
Well, I totally agree.
Nobody wants, sometimes at least in the, you know, I sat on the committee, the joint commission between the US and Canada, you know, for the Great Lakes and other areas. Nobody wants to do monitoring. They think of it as non-sexy. I'm sorry. But, you know, oh, we're just gonna collect all this stuff and it's never gonna come out and there's delays in looking at this and that. And cooperation. Do you have some comments about where the funds should go and come from for that?
I think we need to do a better job of communicating, first of all, to the public. And one of the things I learned at NSF was that I spent a lot of time with having lunch at the Kiwanis Club. I'm not a member, but I would go and speak at the Knights of Columbus, the Better Business Bureau. I was asked to speak to the Women's Garden Club, and I thought, why would I do that? 400 women were there, and they are all over the country. And those were the years I was getting a 15% increase in the NSF budget. So it's, you've got to, we scientists are in our own little bubble, and we talk to each other and we get irate. Why don't they understand us? Because we're not talking to them and telling them. And so I think that's one big problem. The other is that people welcome the fact that we are doing these studies in the Chesapeake Bay. And I spent a lot of time speaking at Tuxent and the Upper Bay and talking to these folks about the work that we're doing, because they want to be informed to make their decision, not to be told what to do, but to figure out for themselves what's best. So I think that's again another chore that we have. By the way, on that Bangladesh study where we went back, we found that not only were the control villages filtering, but we found that within the villages where they continued to filter, those who didn't filter were protected by the herd effect. So if you get, you don't need to have the whole population march stepped, but if you get sufficient, you get the herd effect and then everybody gets protected. Yes, John, I should have.
And we hear in the Pacific Northwest, don't get as many hurricanes. Remember that we may get a magnitude 9 earthquake.
Yes,
I'm curious whether state studies have been launched regarding the event that took place in Bangladesh in 2004, the tsunami, and the activity that took place in Japan, and other major earthquakes and or tsunamis, have there been case studies of pathogen spread in the wake of such events? I think that kind of planning here in the Pacific Northwest, just imagining what could happen and what we could do to respond might be a sense worth our Canadian colleagues.
I think it would be valuable to understand that what is happening with climate change is a direct human health effect. And that it is real, and the infections are very possible. One thing I'd like to emphasize is that the Vibrios are not migrating north. There are papers that are being published that say the infections are they're not migrating. What we're doing, because the Vibrio populations are part of the natural flora, just as E. coli is part of the gut flora. And what we're doing is changing the environment so that they flourish, become abundant, and then become a deadly pathogen, which is the point I wanted to make about Florida. We're picking up Vibrio cholerae, but if we continue with these 90-degree, 100-degree temperatures and the storms that are coming more frequently, we are we are creating the conditions for our own cholera outbreak, possible epidemic. So it's not that I'm saying, Chicken Little, we're going to have a cholera outbreak. I'm just simply saying we're creating the environmental parameters. And it comes back to the question about monitoring the environment. Monitoring the environment is part of public health. And I think if it's understood that it's part of public health, then it would be undertaken. And then that's where it comes to measure, communicating. And I think another flaw that we make as scientists, and I've been talking about this with some of the folks here, so I'm repetitive to them, but to some of you not, is that we scientists work in our own little narrow stream. And I think it's critical for us as physicists, chemists, biologists, when we plan experiments, we should include a behavioral scientist and an economist so that we start from the beginning, if we're doing large-scale studies particularly, what the outcomes will be and how they can be incorporated and communicated, and not wait until the results are there so that you can use the kind of Chicken Little sky is falling approach to tell everybody about it, but in fact, work from the beginning of the large projects with a sociologist, a behavioral scientist, an economist, so that you're already addressing the outcomes that are expected. I know I'm preaching, but this is really important. The other questions? Yes.
You spoke a lot about math. You spoke a lot about mapping. I 100% agree with is an integral part of science communication.
Yes.
Do you think the unique challenge of cholera, mastering the potential impact of cholera, because it's seen as an old-timey disease, for lack of a better phrase, is the highlighting that cholera is still very much a thing that very much impacts the world, kind of high on the list of priorities for the communication of your work and ongoing work?
We did another study that was published. A sociologist did the work, and she did an analysis of whether the population in Bangladesh, because they all have cell phones, they have more cell phones than people on the earth these days. And so the question was, if there were warnings about a cholera outbreak, would you use the cell phone for information? And the answer was clearly yes. So again, it's not that people won't do it, it's that we're not finding ways to reach out and make it happen.
Okay, here's another political circle. So, I was very struck by, the SARS being used to strain water, and
We tested men's t-shirt, doesn't work.
wide underestimation of the women to realize this was an important thing to do. And that kind of bothers me, you know, and like other communities where you didn't test the SARI started using that because they heard about it from the communities that were using the SARI. It's like this underestimation of women, once again, not just as scientists, but as people taking care of health.
I just underestimate, and I have an anecdote to tell. And that is, our work was picked up by BBC, and I was trailed by a BBC photographer. It was on a special program. And we were going to go to MATLAB, where we were doing all the work, but there was flooding, we couldn't get there. So we had to go to a village where we hadn't been doing any work. And we explained to the women what we wanted them to do. We explained that go down, collect the water, and use cloth, and we explained why. And so then the photographer said, okay, we'll set this up. And then he asked the woman to go down and collect the water. And she made it clear that she was not going to collect it without the sari cloth. And that had to be part of the documentary, which it was. It didn't take very long. It took like an instant for her to understand when we explained that you use the sari cloth and look, the water is clear and your kids won't get sick. That was that. The other thing we did, which was quite interesting, was that there were women who were sort of I don't know quite what you call it, the doctors, they have herbs and whatever. We were cutting into their business because people weren't getting sick. So what we did was we paid them to teach the women how to filter the water. Worked very well.
We are at 4:30. Please join me in thanking Dr. Colwell.